All right, welcome everybody. My name is Benji Cohn. Welcome to another episode of the Minnesota Outdoor Skills and Stewardship Series. We are joined today by Doug Dieterman talking about sculpin reintroductions down in southeast Minnesota. So it's going to be an interesting conversation. And Doug, let's just roll right into it.
Sounds good. Sounds good. I'm going to start real quick, acknowledging kind of my colleagues that working with this project. And that's Lauren Miller. He's our statewide fisheries geneticist, and I'm going to cover quite a bit of genetic stuff, and that's all been kind of Lauren's baby. He's really good with that kind of stuff. And I want to acknowledge Brian Byerl. He's fisheries management staff in Lake City. Probably a lot of this sculpin reintroduction program part 2 is kind of his brainchild. When he came on, he showed a strong interest in reinvigorating this program, and that kind of led to a lot of the newer stuff. So quick overview here. I'm going to talk just real briefly about sculpin, and then I'm going to kind of go through some history of kind of how we got in the reintroduction program for this particular fish. That's all this part 1 stuff, and we learned a lot of stuff in part 1, particularly from the genetics aspect. So there was a lot of information that was learned and shared there. And then I'm going to talk about part 2, the main part of this, obviously, and kind of where we are today and where we're kind of going in the future. And so to start with, you know, there's 2 species of sculpin here in southeast Minnesota, the slimy and the mottled. Most of this talk is going to be about the slimy sculpin, and slimies kind of have this whole Arctic distribution. You can see on the map here, they extend anywhere from pretty much all of Alaska out to the eastern seaboard. And if you can, I don't know if you can see my cursor or not, but southeast Minnesota is kind of a little orange blurb here. We really are at the southern edge of the range for this species. And so as a result of that, you know, they are kind of considered a little bit of an indicator species, maybe an indicator of climate change, being a cold water fish at the southern end of their range. A little more biology. They spawn in the spring, usually sometime between late March and late April. The males essentially kind of build this little nest under rocks and invite females to attend them. The fertilized eggs get deposited on the undersides of the rocks, and the male kind of sits there and guards them. They mature at about age 2, and they live about 5 to 6 years total. So kind of a short-lived fish. So how did we get into the reintroduction work? Well, this really starts back, you know, several decades, about the mid-1990s, and kind of came about as a request from our anglers for some more enhanced management for large brown trout. Prior to that, we had all sorts of surveys and some research studies that were kind of focused on increasing abundance of adult browns for anglers, and we were interested in recruitment and stuff like that, but not as much attention was paid to the large brown trout that some anglers actually prefer catching. By large, we're talking like 15, 16 inches and larger. And so what happened was there was a review of our internal information that we had at that time for large trout. That was the staff report 53. And I'm not going to go through all the details of the staff reports, but suffice it to say, one of the big pieces that came out of 53 was how important growth was. And then a few years later, the staff report 57 came out, and that was kind of like a playbook or kind of a recommendations of how we might go about managing a little bit better for larger brown trout. And there were kind of really 4 criteria kind of picked out at that time to help us think about managing for large trout and prioritizing streams. And the first one of those, the main one I'm focusing, that's kind of where this story begins, is the potential for growth. Because the stream actually, is it able to support fast growth of brown trout, and particularly growth to larger sizes. And so kind of one little old chart from those old staff reports is this one. This is a chart that kind of shows a dot. Each one of these dots is a different stream population. And then there's some growth models that we do, these von Bertalanffy growth models. And what those models allow us to do is kind of predict what's the maximum size, or they call it the asymptotic length. But how big can you grow a trout in each one of these streams? And when we started looking at our past data and doing some of these growth models, one of the things we noticed was that there's several streams that, for whatever reason, they just can't grow fish much above 14, 15 inches. And so this really, you know, kind of got to the point of, well, what can we be doing to enhance growth? And so another another table that kind of came out of that staff report 57 was this decision-making key. And the first part of the decision-making key is it asks the question, can fish grow big in this stream? Can they grow bigger than 15 inches from those models? But if they can't grow bigger than 15 inches, then it takes us to step 4. And the next thing it asks is, well, are there sculpin or scuds present? Scuds are a little invertebrate. Their gammarus is the genus name. And both of these are thought to be kind of an energy-rich prey item for trout. And so the thinking at the time was, if we don't have these energy-rich prey items there, perhaps we should be reintroducing them. And so that was kind of the genesis for why we got into the sculpin reintroduction game. So a few years later, 2003, we came up with a sculpin reintroduction plan, kind of had a pretty generic goal of restoring a native cold water fish species and obviously to provide additional prey for trout. The plan acknowledged we had 2 species, mottled and slimy, although to be honest, you can identify them with their pelvic fin rays when they're kind of big, but a lot of streams, a lot of the fish you catch are small. A lot of times we're sampling in, spring or fall. It's cold, your fingers are hurting, and it's really kind of hard to count the fin rays. So a lot of the early reports just noted we had sculpin, and we didn't really know if they were mottled and slimy. One of the things that came later as part of this project is we developed some genetic markers that we could tell the 2 apart, and that's been a big benefit, which I'll talk about in a little bit. But at this point, with the reintroduction plan, they were focused on mostly moving or reintroducing slimy sculpin. And at that time, they identified 3 streams that they called donor streams. These are streams that we thought had enough sculpin that we could pull them out of that stream and then reintroduce them or stock them into a new stream, or what we called recipient streams. There were like 9 to 10 of these. And of course, we had to lay out specific protocols. There's disease testing that had to be done. We had to figure out how we were going to collect them, what time of year we're going to collect them, how are we going to stock them. And part of this plan identified a lot of uncertainties. There's nowhere in the literature that we've seen other people reintroducing sculpin 30 years ago. And to give us some inference, well, how many do we need to stock, for example, to actually start a population? And in particular at this time, there's all sorts of thinking about, genetic concerns. If you don't stock too many, is there inbreeding depression, outbreeding depression, founder effects, all these different genetic things. And so we acknowledge that, yeah, there was a lot of uncertainty, but we're going to go ahead and move forward with some of these reintroductions. And then another uncertainty I'm just going to touch on briefly is, what are the interactions with brown trout and brook trout? Yes, we hope they're going to be an important prey item for larger trout, but if you look in the literature and old reports, there's sometimes been expressed some concerns that maybe they're going to compete with the trout species for different invertebrate prey items, or maybe even the sculpin themselves might actually prey on trout, usually like trout eggs or fry. But there is some uncertainty on what effect are they going to have in the food chain and food web. And so 2003, we started reintroducing sculpin. As I said, we used these 3 source streams. The 9 to 10 reintroduction streams we assumed had sculpin historically, and they'd just been lost over time. This is kind of a quick map to kind of show those. The 3 donor streams are Cold Spring Brook, Beaver Creek, and Garvin Brook. And I'm going to come back to those quite a bit. So hopefully you'll remember Cold Spring, Beaver, and Garvin. And then the triangles are kind of the recipient streams that we put them in at that time. And you can see several were clustered up around Red Wing or Lake City. There's a couple down by Winona, Pickwick, and Little Pickwick. But there was, again, 9 to 10 of these that we put them in. And again, not knowing anything or not knowing exactly how many or how we should be doing this, what we started with was kind of mixing equal numbers of fish from each one of those 3 donor streams. So like in Hay Creek, when I say we put 150 sculpin in Hay Creek, that means we took 50 from Cold Spring Brook, 50 from Beaver Creek, and 50 from Garvin Brook. So equal numbers from each one of those donor streams. In 2004, we had a hard time getting them from Cold Spring Brook. We only got 20 fish when we first started that we put in Gilbert. And so we weren't able to do the 50, 50, 50. So we kind of defaulted to, half from Beaver and half from Garvin. And then in 2005, we were back to our 50, 50, 50. And knowing these proportions that we were putting in kind of became important for some of the genetics work that kind of came in a year or so later. And so what kind of coincidentally happened is there were several researchers with the U of M, some graduate students. David Huff did his PhD up there. Larissa did her master's. Then, of course, Lauren Miller, he wasn't a grad student. He's been affiliated with the University of Minnesota for a long time, and he works for us now. But these three and others, initiated several studies to look at kind of the genetics associated with this reintroduction program. And so I'm going to talk about a couple of these, this proportional ancestry and some of the parentage stuff that they did. I wish I had time to talk about all of it, but I don't have enough time to cover everything that these folks did. So proportional ancestry, again, getting back to it, was key to know what proportion of fish we put in from each one of the three donor streams, from Cold Spring, Beaver Creek, and Garvin. Knowing proportionally what we put in, then we could go in and sample a year or two later and see, well, what's the proportion of the fish that are actually surviving that we put in? And so, is it the same equal proportion from the streams, or is it different? And so I don't want you to get too hung up with the numbers in each one of the cells of this table, but just recognize that the blue cells, those are all genetics of fish that survived better than expected. There were more of them in the population than we expected by the simple proportions of what were put in. And then, of course, the red and the pink indicates ones that did not do quite as well. And so the take-home message here is Beaver Creek, we seem to recover a lot of Beaver Creek genetics in those first few years. And so it suggests that something about Beaver Creek sculpin did really well for survival. Maybe they were more adaptable or something, but they seem to do better in these reintroduction sites than the fish from Cold Spring and Garvin. There were a couple exceptions. You can see Cold Spring, the one kind of bluish cell there. For some reason, Cold Spring fish did really well in Hay Creek, but really not anywhere else. And then fish from Garvin Brook did better in Pickwick and Rock. So a little bit of differences, but the main point was something about Beaver Creek fish looked like they were more adaptable and doing better in these recipient streams. And so one of the other things they looked at was growth, both growth of young of the year, that's age 0 fish, real small ones, and over yearlings, these are like age 1 and 2, so kind of some pretty young adults. And what they have in these charts is their growth rates on the y-axis, and then on the x-axis is kind of what the moms and dads were. So where there you see like a G or a B, that's what we would call a pure strain, meaning both mom and dad came from Garvin Brook, or both mom and dad came from Beaver Creek. And those fish grew pretty good. But once we started getting into the second generation fish, that's the F2, and then the cross, in particular, the crosses between Beaver and Garvin, then we started to see reduced growth. And so this was kind of an indication that, boy, maybe we shouldn't be mixing these fish from multiple streams. The genetic term for this is this kind of hints at outbreeding depression. And so it suggests that maybe our mixing was not necessarily a good idea. Another thing that project that the University of Minnesota staff worked on that was really interesting was this parentage analysis. And so what they did here was in 2007, they ended up stocking some fish from Garvin Brook and Cold Spring into Hay Creek and Rock Creek. They didn't stock them at the same place that we stocked in 2003, 4, and 5. They actually went way upstream, tried to establish a new population. And the nice thing here is they stocked equal numbers. So in Hay Creek, you can see 74 fish, 36 from Garvin, 38 from Cold Spring, but they took a tissue sample from every one of the 74 fish that went into Hay Creek and a tissue sample from every one of the 66 fish that went to Rock Creek. And what this allowed us to do was identify each fish individually. So we knew exactly who was mom and we knew exactly who was dad. And so then they put those fish in, they let them spawn. They came back a year later, fall 2008, started capturing all the sons and daughters from those fish, and they started to put, well, which son and which daughter came from which pair of parents. And they had some pretty interesting results from that. So each one of these bar charts is a family unit. So each bar is a mom and a dad and how many young they produced. And so you can see in that first big black bar graph on the left, that's a mom and a dad. both of Cold Spring origin. Both mom and dad were from Cold Spring, and they produced about 24 offspring. The next bar on that is a different mom and dad, and they produced about, if I remember, 17 offspring. And the black bars are fish in Hay Creek. The gray bars are fish in Rock Creek. And one of the big take-home messages they found from this is that very few moms and dads actually contributed sons and daughters to the population. In fact, it turned out only 19 of the 74 fish in Hay Creek actually produced offspring, and as a consequence produce genetics into the next generation. So most of the fish that we stock, in Hay Creek, that's 74%, never contributed any genetics to the population. And so this really ended up in a loss of genetic diversity because so few parents were actually producing young. And this is typically called founder effects within genetics, and kind of suggested, well, maybe at the numbers we stock, 74, 66, we need to be thinking about stocking a lot more fish than the numbers that we were using. And so there were some other things that those students and staff at the university did that I can't cover, but I will note that one of the things they came up with is a good genetic marker to be able to tell mottled from slimy. And that was actually very useful now in part 2, because now we can take a tissue sample from sculpin that we collect and then send it up to the lab and see if it's a slimy or a sculpin. But they did give us several other important notes. As I said, it's probably shouldn't be mixing sculpin from different populations into the same stream. We should probably be using a sole source ideally, and we should be stocking larger numbers than we had used previously. But there are some concerns about depleting these source populations, right? There's only so many sculpin in there, and we don't want to take too many and hurt that population. So there's a little bit of a balancing act of how many fish can we stock and how do we do this reintroduction work. So this was about 2008, 9, 10. The students graduate. Our staff ended up, we had some turnover, different people coming in, different people leaving. We had other priorities come up. And so after those 9 or 10 streams, part 1 just kind of ended and was kind of left there. So now on to part 2. So about 10 years later, right around COVID, part 2 kind of started. And as I noted, a couple things were the catalyst for this. We had some new staff, particularly Brian Byral, which I noted, came on and had a strong interest in reinvigorating this program. And DNR Fisheries kind of, in a broader sense, kind of was tasked or came the realization that we need to probably be doing more management for non-game fishes. And of course, sculpin are a native non-game fish, so maybe we should be doing a little more management for this. And of course, we had some lessons learned from part 1 that were going to be useful moving forward. So part 2, many of the goals were pretty similar to before: reestablish a native cold water fish in southeast Minnesota, provide increased prey for trout. Some additional justifications, though, is that our streams seem to be improving a lot in southeast Minnesota. We have more miles of cold water fish than we had in the 70s. And as a result of that, a lot of the other prey fishes that used to be present in streams, like fathead minnows and creek chubs, a lot of those minnows and chubs have declined in abundance and in fact have disappeared from a lot of our trout streams. And so now the trout just have essentially various invertebrates or bugs to eat, or else they eat themselves. There isn't much else in there. So certainly, we have more cold water habitat available, and the native sculpin, is perfectly adapted to that niche. So seems like a good time to be moving sculpin around. And then the other part, kind of at the bottom there, our pollution control agency folks, they have several measures that they use to assess, stream health and stream improvement, particularly in the context of some of their programs with the Clean Water Fund to improve streams across the state in southeast Minnesota. And some of these metrics or indices that they use sample fish. And, if you get sculpin in those samples, that tends to score a site pretty high. Well, if we have a cold water stream that's been improving over the last 15, 20 years, but sculpin can't get there, then perhaps they're going to need a little bit of an assistance from us. And so that's another justification for us to kind of move sculpin to new streams. Okay, so part two, kind of 4 big objectives. Again, I don't have time to cover everything, but I'm going to talk a little bit about distribution and distributional changes. Objective 2 was to kind of go back into those 9 reintroduction sites. Recall that in those earlier slide, I said, it's something about Beaver Creek fish seem to do pretty good in those streams. Well, it's been 20 some years since we were in there, or at least 15 years. And so now the question is, it's still a lot of Beaver Creek genetics, or has one of the other two, the Cold Spring Brook fish or the genetics from Garvin Brook fish, started to become a little more dominant? So what's really going on at those sites now, 15 years later or so? And then objective 3 is reintroduce sculpin to new sites. And then 4 is to evaluate food web responses. So historical distribution, one of the things we had to do to kind of get an idea of where we are with sculpin is what was their historical distribution. To know that, you need to know how much cold water habitat you had available. And so what we've used as a proxy for that is essentially our designated trout stream layer. We know there's more cold water habitat than just what's indicated in that layer, but at least gives us a number to work with, and that's about 860 miles. And we assume that sculpin were probably present in all 860 miles prior to European settlement. And then, of course, we had European settlement, we had land use change, all sorts of things happening, stream degradation, and we lost sculpin in a lot of those streams. And so from about 1920 to 2019, about 100 years, we've done a whole bunch of surveys. And so that gives us some idea what the distribution of sculpin is now or has been after European settlement. And so made up kind of a bar chart or a pie chart for that. And what you can see is in all those surveys over 100 years in southeast Minnesota, we've never recovered a sculpin anywhere in about 49% of the stream miles. So we figure sculpin have probably been lost from about half of their original range. There's a small sliver there in red of streams that we've never sampled. These are usually pretty small tributaries. We just can't get everywhere with our resources. But there's been a small sliver of streams we've never looked at. And we've verified sculpin to be present in about 47%. Again, over the last 100 years from 1920 to 2019. And then what I'm going to call our current distribution, the last 5 years, in part because of this program, we've been doing a lot of sampling, some of it targeted to find sculpin and check sculpin sites. Some of it's our routine management surveys, and some of it's just other research surveys. We've sampled over 135 sites. And so now in the last 5 years, what have we shown? Well, we've shown that about 12% of the sites, sculpin were never collected there previously, and we didn't collect them in the last 5 years. So they're definitely not present in 12%. There's 43% of sites of stream miles that we haven't sampled yet in the last 5 years. And so there's, this is going to be an ongoing assessment. This isn't the end all, but there's a good chunk of streams that we still need to check to see if sculpin are in fact still absent there, or if maybe they've recolonized unbeknownst to us. And then about 45% of stream miles, we know there's sculpin. And now, because we have the genetics, we can tell what proportion of those are slimy versus mottled. And that's essentially what this is, the purplish piece of this pie chart. This is about 19% of stream miles we've confirmed have slimy sculpin. The yellow piece of the pie, we've confirmed about 10% have mottled sculpin, and then 11%, the blue piece, these are stream miles that have both species. And it's not always both at the same site. Sometimes it's a stream where we found the slimy in the headwaters and the mottle further downstream, but we know both species are present. And then there's this small sliver, 5%, that we collected sculpin, but for whatever reason, we didn't have the genetics kit with us. The alcohol we had was the wrong alcohol. Well, we weren't able to get a genetic sample and actually determine just a small sliver of streams. And this at least gives us a benchmark to know what our reintroduction program has been doing. And so that's essentially this blue piece, about 10% of the stream miles. Those are sites that we've reintroduced sculpin to. So at least it gives us, again, a little bit of a benchmark and an idea of what contribution we're making for this particular fish. All right, we rechecked those original reintroduction sites, and you can see these are the original 9 sites. Not only were we interested in looking at the genetic changes over the last 15 years, but we just wanted to know, are sculpin even present in these sites or not? It's been, like I said, 15 years since sometimes 20 years we were last in there. And in most of them, you can see we caught good numbers of sculpin, usually well over 100 in the sampling that we did. So it looks like populations persisted in most of those sites, except for 2. One of those is Sugarloaf Creek, which is near Lake City, and we didn't collect any sculpin. We sampled at the original reintroduction site. We sampled upstream, downstream. We couldn't find any sculpin anywhere in that stream. So it looks like that population has winked out. And then there's Latch Creek. We sampled a good portion of Latch. We were able to find 5 fish, but not very, 5 fish is not very many. The one caveat is we were not able to get all the way up in the headwaters at the time. We're still hoping to try and get up there to check, but assuming that this 5 is representative, that population does not look like it's doing very good. So not all of them seems like they've persisted as well as we would have hoped. And then back to this genetics question. So this is kind of a gory little, but colorful little figure. What these are is you can see these little lines. Each one of these little, very narrow lines is an individual fish, and the colors give you kind of the proportion of genetics it has from the original beaver, Cold Spring, or Garvin fish that were put in. And that beaver, Cold Spring, Garvin on the left side, that's the original genetic data. They're colored obviously red, blue, and green. But one of the things you can see is used to be a lot more Beaver Creek genetics in these populations, and that's the red. But now here we are, again, 15 years later or so, and it looks like there's a lot more green. So it looks like maybe the Beaver Creek genetics may not have persisted in the population as long as we thought. It's still there, it's still contributing, but it looks like some of the Garvin genetics are starting to become a little more predominant in a lot of these reintroduction sites. And then, of course, the solid, blue one there, that's probably that Hay Creek site where Cold Spring fish just did really good. So it looks like over the long term, we're seeing some slight changes in the genetics of some of these reintroduction sites. Okay, so objective 3, reintroducing sculpin to new sites. Again, this is kind of one of the main things we're trying to do. Using some of the information that we've obtained from part 1 is that we're trying to stock fish usually from a sole source rather than mixing. And we're trying to usually use the closest stream that we can, but staying within the same watershed. And in fact, we've kind of been informally using the idea of genetic management units as kind of a proxy way to think about each one of the watersheds. And we're generally targeting more than 300 to stock if we can. We're worried about depleting source populations, but we know we're trying to stock more than 300, and we know that it's probably taking multiple years. And you can see in the summary table here the streams that we've been putting them in the last 3-4 years and how many we put in. So like you see Trout Brook, Meesville up by Red Wing, we put 100 fish in there from Hay Creek in 2022. We put in another 88 from Hay Creek in 2024, and again, this will kind of be an ongoing effort. I've also have a couple asterisks here. There's 3 streams that we put them in: Long Creek, Cedar Valley, and Gribbon Creek. And these are part of our assessment for Objective 4 to look at what sculpin are doing within the context of these food webs and what kind of interactions are they having with trout. So I'll move into Objective 4 now. As I said, we're hopeful that these sculpin are going to provide an important prey source for larger adult trout. But there are instances in the scientific literature, both North America and in Europe, that have suggested that sculpin, not always slimy and mottled, there's other species of sculpin, but they can have some other interactions with trout, particularly competition, eating the same invertebrate or bug prey, I guess, if you will, somewhat between sculpin and adult trout, but more often between sculpin and juvenile trout. Excuse me. And then there is some suggestions that sculpin will predate directly on trout eggs or even some of the smaller trout fry. And so, before we go gangbusters and stock sculpin everywhere all the time, we're trying to assess, if there is any potential for some negative interactions here between sculpin and some of the trout species we're trying to manage. And so to look at that, there's a lot of ways to try and assess, predation and competition in particular and niche overlap. And these are big, big concepts in ecology and a lot of debate on the best ways to do them. And we don't have the resources or the time to do everything. So, we're defaulting to, some procedures that we can use to help gain some inference to this. Typically, some of the way you would do this stuff is with diet work, right? You'd pump stomachs of sculpin and trout and see if they're eating the same items or different items. And this, has a lot of, good things about it because you get to identify exactly what the fish are consuming. You have good identification of the invertebrates or fish eggs or whatever they're eating. But there's a lot of drawbacks to this approach. It's very, very time consuming. It takes a long time to sort these samples, to pick the bugs out, to identify them. Sometimes the diet items are all crushed. You're just identifying a mandible or a jawbone essentially from an invertebrate or a leg or something. So it takes a lot of time. And then even the invertebrates particular themselves, it's just a snapshot on the day or two that you collect the stomach samples. And invertebrates are notorious for, being abundant for 1 week and then disappearing, and a different invertebrate item being abundant, hatching out and becoming available to be consumed for the next 2 weeks. So you get real spotty availability of the different prey items. And so, diet work is very useful, but it does have its limitations. So what we've defaulted to is a slightly newer approach to stable isotope analysis. Been around for about 25 years now. And it's another way that allows some inferences to be made in terms of it depicts the food web, which I'm going to show you, and kind of lets you look at which species might be overlapping with each other in terms of their feeding niche space, and what they're consuming. And so I'm not going to go into a whole lot of detail stable isotope analysis. Just suffice it to say there's several of these stable isotopes. Carbon-13, nitrogen-15 are the two biggies that I'm going to focus on. And what happens is, these are elements that are present in various things, in particular, the tissue of different organisms. And as you move through the food chain, the isotopes and their ratios change. And so based on the change in the ratios, you can start to get some ideas of how the food web works. And so the nitrogen-15, that's often used to indicate trophic level. And then carbon-13 is often used to identify the primary energy source. Carbon is like the main currency of energy in a lot of aquatic systems. And so this carbon-13 kinds of lets us pin down where that energy is coming from. And so I'm going to deviate for a quick second here for a slide just to kind of show you the two ends of the spectrum for energy in streams. And so, I got two different pictures here. The one on the left, this is what we would call an in-stream or sunlight-driven energy source, a big open field. The sunlight is penetrating the water column. It's stimulating the growth of aquatic plants and macrophytes and algae. And so really is a sunlight-driven ecosystem. Conversely, you'll get to these, heavily shaded areas that don't have much sunlight. And a lot of these systems, they get a lot of their carbon energy from the leaf litter and the sticks and the leaves that fall in the streams. And so there's a whole bunch of invertebrates that'll feed on the leaves. And although some are eating the leaves directly, a lot of them are eating the bacteria and the diatoms that are colonizing the surface of the leaf. The old analogy they often say is they're eating the peanut butter that sits on the cracker, kind of an idea. But regardless, this leaf litter energy source, carbon, is one of the primary energy sources in streams, and are kind of at two different ends of the spectrum in terms of their carbon-13 values. And so by measuring the carbon-13 in different organisms, you can get an idea of where their ultimate energy source is. And this will make more sense when I get into these next couple figures. And so usually what happens is you get a tissue sample. So like from a fish, we take a small little muscle plug, and then you basically got to dry it down and weigh it and stuff. But you send it off to a lab, and the lab will process it, and it'll come back and tell you what the carbon-13 value is and what the nitrogen-15 value is. And once you have those two, you put them together on a chart that looks like this, This is a general blank one. And so you'll have a particular fish that'll have a certain carbon-13 value, say -35, and it'll have a certain nitrogen-15 value, which is a 3, and it'll appear in the graph at the location of those two intersections. And again, the nitrogen-15 indicates trophic level, carbon-13 indicates energy source, leaf packs versus sunlight driven. And so I'm going to show you some of the data with the food webs here in a little bit. But I'm gonna take a quick second, hopefully to help this illustrate a little better, and I'm gonna focus just on the carbon-13 stuff. And so sometimes you'll see people just look at the carbon-13 energy signature, and that's kind of what I'm gonna show you next. So this is a slide I got from a colleague that's doing some work out in Pactola Reservoir in South Dakota. And he gave a great talk, and I really enjoyed the slides, and it helped illustrate the points. I borrowed a couple slides from him. But what they did is they took tissue samples from several fishes that live in this reservoir, crappie and sunfish and rock bass, and sent it off to the lab and got a carbon-13 signature. And you can see the carbon-13 signature for most of these fishes are kind of in the lower part of this graph. But a couple of them, like golden shiner in particular, the rainbow trout that they stock, have a much different carbon-13 energy signature and fall out much differently on this chart. So they're using a different energy source, ultimately, the rainbow trout are, than these other fishes. And what they were interested here is they were curious of what northern pike were eating. And so they could take a tissue sample from a northern pike and also look at the carbon-13 signature and see, well, do northern pike carbon-13 fall in the lower part of the graph, kind of where all the other crappie and sunfish are, or do they fall much higher in this graph where the rainbow trout are? And that's essentially what this next figure kind of shows. What they did is each one of these dots is the carbon-13 value for an individual northern pike. And then he kind of put this on the size of that fish. So this is a 12-inch northern pike with a carbon-13 value of about -28. And what you can see is there's a whole bunch of northern pike dots for fish that are less than 24 inches, and they're feeding on the bottom end of this chart, which suggests they're feeding on those perch and bluegill and crappie. But once they get above about 24 inches, then their carbon-13 value changes a lot. They're feeding much different energy source, and it corresponds a lot with where those rainbow trout are. So it's a good bet that these bigger northern pike are eating a lot of those rainbow trout that are getting stocked in the reservoir. So I really like this example, and I'm going to show you a little bit of the same type of information with the data we have. So again, real quick, so the study design here is to assess these food web changes. We have essentially 3 pairs of sites. Middle Creek and Long Creek are a pair, Pleasant Valley and Cedar Valley, and then Big Springs and Gribben. And essentially these are streams kind of clustered. The pairs of streams are really located close to each other. And we collected food webs in all 6 of these streams. And then we stocked sculpin after we collected food web data in 2022. And so we've got pre-sculpin introduction food webs in both a treatment and a control. We've stocked sculpin. We're waiting for sculpin to colonize and become established in a population. We'll repeat the food web, and we're going to see how it changes now that we've introduced sculpin. But I'll give you a little peek of what the food webs look like before we stock sculpin, just because I assume most people are interested in it. So this is Gribben Creek food web. The carbon-13 values for essentially the bottom of the food chain. So the 3 bottom ones here are watercress and ranunculus. Those are aquatic plants, and filamentous algae. So this is the sunlight-driven part of this food web on the bottom. A little bit higher up on the carbon-13, this is where the leaf packs are and all the carbon energy that they have. And so then the curious part is, where do the invertebrates fall out? And you can see most of them are falling pretty low on the graph, probably feeding on the watercress and ranunculus and ultimately getting their energy source from sunlight. One of the ones that sticks out is these little snails, and they're feeding much differently than most of the other invertebrates. They're probably up here feeding a lot on the leaf packs, again, probably scraping off those bacteria and diatoms, that peanut butter on the cracker analogy. And so they are definitely have a different signature. And then what we're interested in is, where are the trout in this? Well, we've got 2 size groups. We've got juvenile trout, or small ones, and they're tending to feed again, pretty low on this graph, probably ultimately getting their energy from the sun. But the larger brown trout, they're up a little bit higher, and they're probably getting a mix of energy ultimately from the sunlight, but they're also probably getting a lot of it from the leaf packs through the snails that they're consuming. The trout aren't eating the leaves, they're eating the snails that are eating the leaves. But it kind of shows where they exist in this space. And so then to put the carbon-13 values in conjunction with the nitrogen 15 trophic level, this is kind of really where we're trying to get. This is the kind of figure that you get. So now nitrogen 13 energy sources on the bottom line here, the bottom axis, and trophic levels on the y-axis on the left-hand side. And you can see at the bottom of the food chain, we have leaf pack, which is the dark green circle, and then the filamentous algae is a little bit lighter green. And of course, a little bit above them, trophic level wise, are a bunch of invertebrates. Some of these caddisflies, the gammarus or scuds, if you will, are that little red blob. And then, of course, the gray blob right above the picture of the snail, that's where the snails fall out. And then, of course, another trophic level higher is the juvenile trout, again, feeding on that ultimate sunlight driven part of the energy source. And then the bigger brown trout in that dark blue ellipse, if you will, which is again probably a mix of sunlight but also some leaf pack and ultimate energy source. And so this is really kind of show us the feeding niche or the feeding ecology of where these organisms are fitting in this food web of Gribbon Creek. And again, you know, we don't really have good direct diet data here. This is, we're getting some inferences based on where they're positioned in this graph. But as I alluded to, luckily, we had some good diet data collected by some students from the U of M. And so I've kind of cherry picked one of their figures. This is some data from Will French, who did his PhD at the U of M, and he did a bunch of diet work on trout at a bunch of different streams. And the second stream from the left, you can see is Gribbon Creek, and you can see this little black hash mark bar. And so that's actually all the snails. And so what the take home point here is Will French in his diet work found that, yep, brown trout in Gribbon Creek were eating a lot of snails. And this corresponds very nicely with the graph I just showed you, that those brown trout, particularly the bigger adults, are feeding on that part of the graph with snails and ultimately a leaf pack energy source. Okay, I'm going to go with one more here real quick. This is Middle Creek Food Web. Again, here's where the base of the food chain is, the different plants and algae with leaf packs at the top. Here's where the invertebrates fall. These are tending to feed a little bit differently, a little bit more on the watercress, and then much more on the leaf pack. Obviously, snails are way at the top again at the leaf pack, but now the gammarus and that black kind of half circle thing is a limnophilid caddisfly. They're feeding a lot on the leaf pack stuff. So a little different food web composition here. Here's where the brown trout, the juveniles and the large adults. But the interesting point about Middle Creek is this stream also has native brook trout. And so we collected tissue samples from them. And it looks like they're feeding fairly similarly to what the larger adult brown trout are. So it looks like there's probably quite a bit of diet item overlap between these bigger browns and these brook trout. And again, to kind of put this together in this figure of carbon-13 energy source on the bottom line and trophic level on the y-axis is kind of what this graph looks like. And so, you know, the leaf pack is down at the bottom, the dark green, you got the invertebrates in the red and orange. But the key point here is the juvenile brown trout in light blue, those bigger brown trout in dark blue, and then all the red dots are all the brook trout. And so not only are the brook trout feeding on the same ultimate energy source as the adult browns, but they're feeding at the same trophic level. So this is pretty strong evidence that they are using the same diet items. Now, whether it's competition or not, that's hard to determine just from this, but certainly looks like a lot of overlap between native brook trout and brown trout in Middle Creek. And so lastly, kind of try and bring it all home, where we're going with the food web plots. I've got kind of hypothetical data here. So we've kind of got all this pre-sculpin data. We've got these ellipses drawn on these previous charts I showed you. So we know where the juvenile brown trout, we know where the adults, we know where the brook trout, we know where the basic invertebrates are. You know, the question is going to be, what happens when we put sculpin in? Where do the sculpin fall in these charts? And not only where they fall, excuse me, where do they, does that have any, does that have any bearing, or does that change where the brown trout and where the brook trout are? I talked about, the scientific literature suggests, maybe that sculpin are potentially competing with juvenile trout. If that's the case, then we're going to expect to see our sculpin ellipse overlap our juvenile brown trout ellipse, and kind of the same thing with brook trout. So, all we have at the present is just, the pre-sculpin introduction food web data. We've stocked the sculpin. We'll repeat the food webs in a few years and really kind of look for the changes there. And I know that that's a lot. Hopefully that was kind of entertaining for you. That's pretty much what I have. And if anybody's got any questions, I'll do my best to answer them.
I think that was fantastic. It was great talk with a lot of information. So I appreciate your taking your time to share that and some of the research, and try to go back and see if I can figure out how to tie a leafy fly with a snail on it to see if I can catch some bigger trout. I don't know if I can be part of your study or not, but we'll try. If anybody does have any questions, please feel free to put them in the Q&A in the Slido part. Again, bottom right-hand corner, those three ellipses, open Slido and the Q&A there. I got one question from Kelly here. Was the Vermilion River looked at regarding growth potential?
That's a good question. The short answer is no. The Vermilion is way up, way up by the Twin Cities, and it's not really in our typical Southeast trout management area, which is, Lake City, Lanesboro, Iowa border. And so we were looking mostly at streams down there. And that's a good question, Kelly, because, when a lot of that stuff was being compiled in the mid to late 1990s, I'm not even sure how much trout information they had collected. I mean, I know the Vermilion is, got kind of a reputation for producing some nice sized fish. And I know that the East Metro office does a lot with the Vermilion River system now, but I don't know how much they did in the early to mid 90s. That's a good question. I might have to follow up and ask them. But certainly, to answer your question, no, it wasn't looked at for growth potential with this particular set of studies.
A very interesting river system. I know our local TU chapter has done a lot of habitat work down there, and there's a lot of really great work that's been done to help preserve that, and some good fishing down there for sure. So Kelly put a comment in there about connecting with you. So Kelly, please reach out to Doug. I don't know if you want to, you can throw do you want to share your email address, Doug?
Sure.
We could throw that in the chat maybe.
Yeah.
And do that. If anybody else has any other questions, please put them in the Q&A section there. And if you're interested in trout, we're going to shift gears a little bit here. Our next webinar in 2 weeks, right before Thanksgiving, is number 229, and we're going to be talking about fall lake trout stocking. So Mario from West Metro Fisheries is going to join us and talk about stocking some lake trout in area lakes specifically for ice fishing for trout. So should be a good conversation also.
And certainly if people have questions, after the fact, that my email there, feel free to reach out to me.
Right. And again, that is in the chat function there. So don't see any other questions. If you want to stop your PowerPoint there, Doug, you can do that.
Okay.
I don't see, you must have answered everybody's questions. I don't see any other questions in there. So, and it's such a nice day outside in front of the pending winter coming here. It looks like this weekend it might get a little rain and snow. And it's deer opener this weekend, so I'm sure a lot of people are out getting stuff buttoned up and ready for hunting season and the hard freeze that's coming. So I appreciate everybody that joined us live in person today. Thank you for doing that. Hopefully you can get out and enjoy a little bit of this great weather we have, and hopefully they can get out, take somebody out new hunting or fishing this weekend. So it should be a decent weekend for that. So see you in a couple of weeks for our fall trout lake stocking webinar. Until then, stay safe and enjoy the outdoors.